Okay, we are going to start the cell today, and we'll be talking about functions, the various parts of the cell, the organelles which we see. We'll also talk about cell transport mechanism and then cell division. So first let's look at the functions of a cell. So one is it maintains boundaries by means of its plasma membrane. A cell metabolizes, it carries out a lot of the functions which we talked of, which were functions of a living organism. For example, metabolizing, after it has digested some food, then it metabolizes that. It then gets rid of waste by excretion. Most cells are capable of reproduction. There are some cells in the body which are living cells but do not reproduce. One very good example is the red blood cell. So it's living, but it doesn't reproduce. It has a limited lifespan of 120 days. Also, when cells, the more highly evolved cells get, the less capable they are of reproducing. So, for example, we'll see when we do tissues, the different types of cells. There are 4 different types of cells. In fact, when we did introduction, we talked about it. There were epithelial cells, muscle cells, connective tissue, and nerve cells. Now, muscle cells and nerve cells are highly evolved, and so they don't reproduce. If you have a problem in the brain, if there's a part of the brain that's damaged, the neurons in the brain will not reproduce. They will not regenerate. So, they died then and there. So, evolvement is at the cost of reproduction. Some highly evolved cells, like the nerves, they're capable of doing so many things, but reproduction is not one of them. So in fetal life they do, but once the child is born, and in adult's life these cells don't reproduce. And red blood cells don't reproduce because they do not have a nucleus, so there's no nucleus in them, which tells you that the nucleus, which is part of the cell, is very important for reproduction. Also, cells are capable of movement. The movement may be very slight and you cannot observe it, or the movement may be observable movement. So, but they are capable of movement, and usually this movement is shown whenever a stimulus is, a cell is stimulated, and then that cell gets irritable. So that's that response to the stimulus, which you see as irritability. And most of the time that irritability then is shown as some sort of movement that occurs. Now, there's some cells in the body which are extremely rapidly dividing cells. Can you think of some parts of the body where the cells are very rapidly dividing? Skin, and actually when you say skin, you really mean it's only the top layer of the skin, right? And this is known as epithelium. So epithelial cells which form the lining. So, for example, the epidermis of the skin, which is a type of epithelium, then the lining of our organs like our mouth cavity, our gastrointestinal tract, our respiratory tract, all the lining inside, that's all epithelium, epithelial cells. So those are rapidly dividing. Anywhere else? Pardon? Hair, very good. Yes, hair follicles, excellent. Hair follicles. And another area in the body is bone marrow. Okay, so these are inside the bone marrow, the various blood cells are produced and then pushed out into the bloodstream, very rapidly dividing cells. This is the reason why, and here's why I'm telling you this, is that from now on, whenever you kind of look at commercials which have something to do with medicine, always ask yourself, why is that happening? Because, you start getting some knowledge right here. And then you should be able to kind of relate what's happening up there, what knowledge they're giving you and what you're learning here. So to give you an example, you may have seen commercials which show somebody undergoing chemotherapy. Right? I mean, there's one which comes up about a grandfather who undergoes chemotherapy, and one of the complaints that he has is he's extremely fatigued, very, very tired. And you may have heard of or seen people who have chemotherapy. What happens to their hair? Falls off, right? The hair falls off. And people with who undergo chemotherapy also are prone to nausea. They tend to have a lot of vomiting. So doctors have to give them something in order to alleviate this vomiting. Now, here's why I'm trying to kind of connect the dots. Cancerous cells are cells which divide very, very rapidly. But the division is an uncontrolled type of division. It's not a programmed division. It's not something that the body needs to do. So it's very uncontrolled. So you get these huge, big lumps called tumors which form all over the body. So when you give chemotherapeutic drugs, what do you want to do? You want to destroy these rapidly dividing cells and get rid of the cancer, which is what they do. So chemotherapeutic drugs aim for rapidly dividing cells and they kill off the cancer cells. But the side effect is that they don't know how to choose between rapidly dividing cells which are not good for the body and ones which divide rapidly as a normal process in the body. So one of the side effects is that they affect epithelium. So, which is why like a person has nausea, because they affect the epithelium of the gastrointestinal tract. And so the lining doesn't regenerate as fast as it should. So the person tends to have vomiting and tends to have nausea. It affects hair follicles, where again the rapidly dividing cells are seen. So that's why the hair begins to fall off. And the reason a person undergoing chemotherapy is extremely anemic and very, very fatigued is because the bone marrow is affected as a side effect. So when the bone marrow is affected, the red blood cells, white blood cells are not formed the way they should or as fast as they should. So they have less red blood cells in their body and they hence end up with anemia. So often one has to give them blood transfusions or other things to kind of help them with this kind of side effect. So now do you see why chemotherapeutic drugs would affect rapidly dividing cells? And these are normal cells in the body, but this is a side effect. Okay? So here let's look at the parts of a typical cell. The outer layer is formed by a membrane which has pores, tiny pores, and this is known as the cell membrane or also known as the plasma membrane. Inside it contains a jelly-like substance, fluid jelly-like, which is known as the cytoplasm. And in this cytoplasm is another area where there is a condensation of DNA, and this is called the nucleus. So this area is called the nucleus, which is also contained within a membrane which has pores, and this is called the nuclear membrane. And seen within the nucleus here is the nucleolus. So within the nucleus is the nucleolus. So we'll describe each one of these areas in turn. So let's look at the cell membrane or the plasma membrane. So when you look at it from the top, the plasma membrane, notice how it kind of looks like a little mosaic. You've got these round heads and then you've got this thing bulging over there, then something sticking out. So when seen from the top, it looks like a mosaic. And also in the plasma membrane, the proteins which are present inside and the fat, cholesterol, which is the fat, it's able to kind of move around. So therefore the plasma membrane is fluid and looks like a mosaic. So usually we call it a fluid mosaic model for these two reasons. Looks like a mosaic when you're looking at it from the top, and fluid because these proteins and the cholesterol and all are able to kind of move about within the plasma membrane. Now when you look at this plasma membrane, notice it is a double layer of phospholipid. Phospholipid means fat with a phosphate molecule. So fat with a phosphate molecule. So there is a double layer. So this is one layer and this is the second layer. And in between these two layers, you'll find in places proteins are kind of scattered throughout. So all these purple things are proteins which are present inside. Some of them may pass right through and through the membrane. Some may be attached only to one end of the membrane, either the outer or the inner, and we'll discuss these proteins later. Now when you look at this double layer, and here's why I want you all to always pay attention to these pictures. When you look at this, notice that these rounded heads are called polar heads of the phospholipid molecules. And up here, when you see these are called the non-polar heads. Remember, water is a polar molecule, right? We did that in chemistry. Water is a polar molecule. Outside the cell, there's always, which is the area outside the cell is called extracellular area, and that's got fluid. Inside the cell here is cytoplasm, which is also fluid in nature and has water in it. So therefore, on either side, you can see interacting with the outside and interacting with the cytoplasm. You need these polar heads so that they will be able to interact with the water which is present outside and which is present inside the cell. Okay? So these polar heads are also known as hydrophilic, meaning water-loving. Water-loving. The non-polar tails are called hydrophobic, meaning hating water. Hydrophobic. Now, in between this double layer of phospholipid, we have cholesterol, which is a fat, a type of fat. You can see cholesterol present here. These yellow things are cholesterol. And this cholesterol lends stability to the cell membrane. The reason you have cholesterol present here is it gives stability to the cell membrane. By that I mean that if it becomes too cold, this is phospholipid, right? What happens to fat when things become very cold? It tends to solidify. And what happens when it's too hot? It tends to liquefy. This cholesterol makes sure that the cell membrane remains stable in all temperatures. So it prevents it from solidifying in too cold a temperature and prevents it from becoming too fluid in a hot temperature. Okay? So it lends stability to the cell membrane. Then we have proteins which are present, as you can see up here, proteins which are present in the cell membrane. These proteins may be present through and through, you can see up here. So they're right through the cell membrane, both at the outer and inner end. So they'll touch the extracellular fluid outside and be in contact with the cytoplasm inside. They may have little channels in them. Notice these channels which are present up here. See this? They may have channels present in them. So such proteins which are more or less present through and through the cell membrane are known as integral proteins. And then we may have other proteins which either sit like this on top of an integral protein up here. So they may be only on the outside, or they may be only present inside towards the cytoplasm, or they may be only present on the outside towards the extracellular surface. Such proteins are naturally called peripheral proteins. Peripheral meaning towards the towards the periphery, either end. Yes. Most of them go all the way through, at least 2/3 of the way through. Most of them go all the way through, or 2/3 of the way through. Okay. The function of these proteins is, one, they act as carrier proteins, so they help to transport substances. These proteins may also have a carbohydrate molecule attached to them, so then in that case they'll be called glycoproteins. They usually are also important for cell-to-cell signaling. Some act as receptors. They also keep cells together. So there are lots of functions of these proteins. So cell-to-cell signaling, they act as channel proteins. They allow they help substances pass through. They may act as receptors in some areas. And then here I showed you that there is a glyco, means from the word glucose. So this is to do with carbohydrates. So a carbohydrate molecule may be attached to a protein, or a carbohydrate molecule may be attached to the fat part, so then it's called the glycolipid. So these glycocomplexes are also very important in cell-to-cell signaling, differentiating self from non-self. Like the reason your body doesn't attack its own cells is because you're able to recognize self. But something foreign comes into your body immediately arms itself to destroy that. So that's non-self, not belonging to you. So allows you to recognize self and non-self. So cell recognition. Okay, so these glycocomplexes are important. There are lots of other functions. These are some important functions of these parts of the cell. Now, having looked at this picture, tell me this question. Okay, well, most of you got it right. Yes, it's the polar heads face the exterior and interior of the cell with the tails forming the center of the membrane. And here's why I tell you always to look at a cell at the slide really, really carefully. Notice this. So look at the labels. Look at these polar heads. Can you see these polar heads towards the outside? Polar heads towards the inside of the cell. So they are both towards the exterior and the interior. And what do the tails do? The tails kind of form the middle part. See this up here? So therefore, this with the tails forming the center of the membrane. So that's why this was the correct answer. So always kind of look at these labels very carefully because it will really help you answer a lot of questions. Now, here's areas that we'll see how these proteins are used. So cells, when one cell comes close to another cell, it's not always that these cells are very tightly adherent to each other, and neither is it that the cells are very loose and can just be pushed apart. So they form junctions between the plasma membrane of one cell and the plasma membrane of the cell next to it. And these are known as membrane junctions. And actually, if you look up here, all the functions are really given very close, just below it. So the first type of cell membrane junction we see is what is seen here is what is known as tight junction. As the name suggests, tight junctions means the cells are tightly adherent to each other and they prevent anything from passing from the extracellular fluid into the cell. So if you look up here, so this is, imagine there's one cell here like this and this is another cell close to it. If there is fluid present outside, this fluid, if there was a space, this fluid can pass through and can enter the cell, right? Now, whenever the junctions between these two cells are really tight, it will prevent things from the outside getting into the cell and also prevent leakage from here outside. A good example is in the gastrointestinal tract. You eat all kinds of food, so the lumen or the cavity of your stomach and your intestines contains all kinds of stuff, some good, some bad. You might have enzymes inside which are produced, which are meant to digest food stuff. And those enzymes, if they were able to come back in and go into the cell, they might cause digestion of the cells. Or you may have pathogens inside, that means viruses and other things, which also, if they were allowed to pass in through, they could get into the cell and they could destroy the cell. So you want to prevent that. So this is kind of a safeguard mechanism. So this kind of tight junction is seen, and it's because of the adherence of proteins. This kind of tight junction is seen in the gastrointestinal tract, one primary area where you see that. The second type of junction is what is called desmosomes. And this is usually seen in the skin, where what happens is that between two cells, we have protein filaments which pass across, and they kind of bridge the gap between two cells. Like you can see up here, there's a sort of a thick area on this side of the membrane, a thick area on the other side, and it's the filaments, protein filaments pass through. So these are called plaques. They are like anchors on either end. And the idea is that they keep all these cells together, making the cell look like a sheet. So they kind of are present where cells are arranged like a sheet, like for example in the skin. And they kind of withstand stress because they kind of unify all the cells together, making it like a sheet. And they prevent the cells from tearing. Okay, the reason if you just press your skin doesn't tear immediately. It's because of having these kind of things called desmosomes. When you look histologically at the skin, you will find that these desmosomes show up between cells as little. So if this is one cell in the skin and this is another cell, you actually can see little prickly things up here. Because histologically, when we look at a slide and we process the slide and then we make sections of it, we use alcohol to dehydrate the cell and then we have to stain it. So when we use alcohol and we dehydrate the cell and the cell shrinks like that, the desmosomes actually show up as little spines on the outside of the cell. So this layer of skin is actually called the spiny cell layer, or it's called stratum spinosum because of this spiny appearance, because the desmosomes are seen like that. Okay? So the purpose of desmosomes is to unite cells as a sheet, withstand stress, and prevent that area from tearing very easily. The skin is one particular area where you see desmosomes. The third is where you want actually passage of substances between cells because you want things to happen really fast. For example, conduction of impulses, like in the heart muscle or smooth muscle. When impulse reaches one heart muscle, it should pass on real quickly to another one. So it passes really quickly, and that's because of junctions known as gap junctions. So there are gaps between the two cells, which you can see here. The gaps are like channels. So they allow the message to pass across from one cell to the one next to it and then speed up the impulses. So this is very typical of heart muscle and smooth muscle, this kind of gap junctions. So these are the three main types of junctions which are present between the cells and between the cell membrane of one cell and the one next to it. Smooth muscle, heart and smooth muscle. So let's now look at what are some of the functions and properties of plasma membrane. One property is that to be permeable means to allow things to pass through. When we say something is impermeable, that means it doesn't allow anything to pass through. When we say something is selectively permeable, like the plasma membrane, it means that it allows only certain substances to pass through, not others. So which substances does it allow? It allows substances which have very small in size. So really tiny particles can pass through. So ions tend to pass through very easily because they're quite small. Gases can pass through very easily. It also allows, if there are charged molecules, so ions, for example, sodium has a positive charge. When we were doing sodium and chloride and we got the two together, remember sodium gave off an electron, so became positively charged. Chloride received the electron, so became negatively charged. So these were called cations and anions, remember, also called electrolytes. So if molecule is charged, where it's called an ion or it is called an electrolyte, that also passes through very easily. It just so happens usually charged molecules are rather small. Also, if the substance that has to pass through is soluble in fat, it makes it much easier for it to pass through because the cell membrane is made up of phospholipid fat, right? So if that substance can dissolve in fat, it will pass through very easily. So the substances should be lipid soluble. So this is one property of plasma membrane. Anything that's very large, that means it has a high molecular weight, will find it difficult to pass through. So it has to use some other mechanism if it has to get into the cell. Small substances are able to pass through real easily. If they are charged, again, it helps them to get across. And if they are lipid soluble, they can pass through the membrane easily, and that's how it's selectively permeable. Amongst functions, just having the membrane on the outside is protective. It gives shape. It also allows substances to pass in and out, so it helps with transport, and we'll be seeing that when we do cell transport. And then in cells like the muscle and nerve cells, we have something called the resting membrane potential. That means when we look at, let me just draw a muscle cell. A muscle cell is not a small cell like this, a skeletal muscle cell, but it's kind of like a long cylinder. And in a muscle cell, on the outside of the cell are many sodium ions, and sodium is positively charged. So there are many sodium ions. Inside the cell are potassium ions, which are also positively charged, but there are more sodium on the outside as compared to the potassium on the inside. So we say that the outside of the cell membrane is positively charged compared to the inside. This kind of a charge that exists is known as the resting membrane potential. When we do the muscular system, I will talk about it in more detail, but for now just kind of try to understand. This is something that is there in the body, so when your muscles are at rest, we have a lot more positive charges on the outside compared to the inside of the cell, and we say that the outside of the cell is positively charged compared to the inside, and we call this the resting membrane potential. When a nerve impulse comes, and it comes here, what it does is it kind of disturbs the permeability of this cell membrane. It allows sodium ions to pass into the cell, and after some time potassium starts coming out of the cell. Okay, so can you see that this resting membrane potential has been disturbed, and then this allows the impulse to spread further. It sort of goes on, and then a lot of things happen inside the muscle, and that's how your muscles contract. Then after some time, the sodium has to be pushed out of the cell and the potassium has to be brought back into the cell in order to come back to that resting membrane potential. And that pushing the sodium out and bringing the potassium inside is one means of transport that we will see when we do later. So that involves this transport of ions. Also, the plasma membrane, because it's the boundary between what is inside the cell and what is outside, it helps the cell interact with the environment which is on the outside. Because of the presence of those proteins which are there, cholesterol, those glycocomplexes, so you know the cell-to-cell interaction or cell-to-environment interaction is all done because of the presence of this plasma membrane. In certain parts of the body, the plasma membrane gets highly specialized in order to carry out some functions. And three major specializations are seen. The first one is what is known as microvilli. The word villus means a projection. So let's take a cell which is resting on a membrane called the basement membrane, and this is a simple cell like this with a nucleus. And let's take another cell which is here, and this cell now actually throws the top surface into little folds like this. These little folds which you see, this is known as microvilli. So these are surface projections. When you look at it under the regular microscope which you use in the lab, the light microscope, you cannot actually see the infolding so clearly. What you will see is, you will see a cell like this, and you will just see a very, very fuzzy appearance on the top. It looks like a brush. So this is often known as brush border. So brush border, so microvilli, which are surface, this kind of folding, this is the surface folding, or what is also known as invaginations, where the cell membrane has kind of folded upon itself like that and gone in. These, and so you can see these in between, you can see these little projections. These invaginations, when you look at it under the electron microscope, you can actually see that clearly. But when you look at it under light microscope, which you see which you use in your lab, you don't see this infolding that clearly. What you just see is the top area looks a little fuzzy like that, and it looks like a brush. So we call it brush border. But what basically it is this kind of thing. This is seen, where do you think you will see this kind of microvilli? Where would you need this? What would be the function, in other words, of microvilli? Increase surface area. So remember I showed you here you had this one cell, and here in that same surface area, notice how I made this. So can you see that it will increase the surface area? Okay? So the function of microvilli is to increase surface area, and that surface area is for absorption and also maybe for secretion. The more surface area you have, the more you can pass out as well. So absorption and secretion. So it's seen in those areas where a lot of this goes on. For example, the gastrointestinal tract, because you absorb nutrients, you also secrete a lot of enzymes into the tract. Okay? So microvilli are seen in the gastrointestinal tract. One really good area where you see them. You also see them in kidney tubules in some parts, again because of absorption. The second type of specialization we see is what is known as cilia. Now here notice the difference. Cilia are thin hair-like projections from the cell. So if this is the cell, it's very thin hair-like projections out of the cell. Can you see the difference between cilia and microvilli? Right? Okay? So what do you think would be the function of cilia? Thin hair-like projections. Movement, exactly. Where you want, because this hair-like, all of these hairs, they can kind of beat in one direction and cause movement of substances which are traveling over them, like fluid or air or things like that. So cilia are seen in the respiratory tract, so that they can, for example, in your nasal cavity, the cilia beat towards your throat, towards the back of your throat. You cannot see them. If you peep inside your nasal cavity, you cannot see cilia. Remember, this is microscopic, okay? If you looked under a microscope, you will definitely see cilia. A good, well-stained slide, and when you'll look at one type of epithelium, you'll be seeing these cilia. So they beat in your nose, in the nasal cavity, they beat towards the throat so that any cell debris that you have taken in along with the air that you breathe goes into your throat and you swallow it. Now suppose that goes into your respiratory tract, into your trachea and all that, you don't want it to go into your lungs. So in the trachea and the rest of the larynx, all the cilia which are present, they beat in the opposite direction, upwards towards the throat, so that you can cough that out. So do you see the purpose of cilia? Again, for movement. The third type of specialization we see is what is called a flagellum. Flagella are plural, flagellum is singular. And this is seen in the sperm. In humans, it's seen in the sperm, and it's just one thin tail-like structure which is present. So this is called the flagellum. It is single. If it's normal, it should only be single. And the function is to give motility to the sperms, because the sperms are deposited in the female genital tract and they have to swim all the way up to the uterine tubes in order to meet the ovum. So they have a long way to go. So they need something to help propel them. So this is for movement of the actual cell, and it causes the cell to move forward. Whereas in cilia, that doesn't make the cell move, but it kind of sets up currents, either air or fluid currents, in order to move substances. Okay? So let's answer this question. So we have this area of the body which is subject to a high degree of mechanical stress. So what type of membrane junctions do you think will be present? Desmosomes, yes, desmosomes. Gap junctions are to allow fluid or other substances to pass between cells. Tight junctions is to prevent substances from entering into the cell from the extracellular fluid. Remember, desmosomes are what keep cells together. So they allow the cells to act like a sheet. They kind of attach cells to each other, preventing them from stretching and tearing. So next is, one of the functions of the plasma membrane was transport of substances. So substances move across the plasma membrane, and again the membrane was selective. It didn't allow every substance to move. Remember, size, molecular weight, charge and solubility were important. So now there are two ways that substances can go across the cell membrane. One is without utilizing energy, when we call it a passive process. So passive, all in our case, when we talk of membrane transport, always means that it is without the use of energy. It might use some other mechanism to get into the cell. It may take the help of something else, but it does not use energy. And what did we say was the energy molecule that a cell uses in order to carry out its function? ATP. So that means without use of ATP. When I say energy, I'm saying without use of ATP. So that's one. Second is, a passive process always occurs along a concentration gradient or down a concentration or pressure gradient. By that I mean, it always goes from an area of high concentration to an area of low concentration, or from an area of high pressure to an area of low pressure. Okay? So along or down a concentration gradient, meaning goes from an area of high to an area of low concentration or high to low pressure. So if the pressure is very high, for example, in one artery and less in the other, the blood will flow from the area of the high pressure to the area of low pressure. Under passive process, there are various types that we have of passive processes. The first one is called diffusion, or just simple diffusion. This occurs along a concentration gradient. That means it goes from an area of where it's highly concentrated to an area where it's not so concentrated, and usually takes place where gases have to go from an area of high to low concentration, or we have lipid soluble substances, so they can pass through the cell membrane. Lipid soluble substances. So if you have too much sodium on one side of the cell membrane, less on the other side, it will go from where it is more through the plasma membrane into the part where it's less. Same with gases. And here's an example, if you pay attention. In your lungs, so in your lungs, this is an air sac in your lungs, and when you breathe in, okay, and air goes down your trachea and it gets into the lungs, right? So you breathe in oxygen. So oxygen goes in, and when it comes into the lung, there's a lot more oxygen inside the lung. And what do you breathe out? It's carbon dioxide. So carbon dioxide goes out in the opposite direction, right? And that has to come from somewhere. That carbon dioxide is produced in your body, in your cells, and it travels through the blood. So in the lungs, it's lying in a blood vessel up here. So inside this blood vessel is CO2. This membrane between the lung air sac and the blood vessel is very, very thin. So because oxygen is more here and less here, oxygen diffuses into the capillary. Because carbon dioxide is more inside the capillary, less in the alveolus or air sac, it goes into the lung, and that's why you breathe it out. This oxygen which you take in gets into the blood vessel, and from there then it's carried to all the tissues of the body. So can you see how gases go from an area of high to an area of low concentration? Okay? So that's just called simple diffusion. The second is what is called osmosis. Osmosis is diffusion of water. So it is diffusion of water, and we assume that anything else which is present is not able to diffuse. So we did this when we were doing chemistry, so let me go over it again. So pay attention over here. So let us say that we have two beakers up here. So here's one beaker and here's the other beaker up here. And in this I put in 50 ml of water. Okay. So what is the concentration of water in this? 100%, right? It's all water. So 100% concentration. In this one, let's say, for example, I had 50 ml of water and then I added 25 grams of salt, sodium chloride. When sodium chloride goes inside, and I add that sodium chloride up here, so my volume now becomes 75 ml, okay? And sodium and chloride will break up into sodium and chloride ions like this, okay? How much water is there? 50 over 75. Do you see that? Right? So this is 75%. Water, the amount of water is only 75%. Do you see that? These sodium and chloride which have gone inside and have kind of broken into sodium ions and chloride ions, these are what are called solutes. And they are dissolved in this water which is called the solvent. Followed? Okay. How much, what is the percentage of solutes in this? 25%, right? 25 over 75, 20, or whatever the calculation, I may be calculating it a little wrong, 33% I think. But anyway, okay, so you can see the solutes is this much. If you compare these two solutions, do you understand that this solution has no solutes at all? This solution has some solutes, okay? If we take a third solution up here, and there was 50 ml of water, and let's say I added 50 grams of sodium chloride. Now what will happen? 50% is sodium chloride, 50% is water. Do you see that there are more solutes in this one compared to this and this, right? So this has most solutes, okay? So whenever in water you have substances dissolved in it, which are known as solutes, we come up with something known as the tonicity. So the tonicity is the amount of solutes which are present in a solution, and because of the amount they can change the shape of the cell. So when we compare these two solutions, or we compare these two solutions, this solution, so let me compare solution if I call A, B and C. If I compare solution A to B, this has no solutes at all. This has some amount of solutes. So I can say that solution A is hypotonic compared to solution B, because it has, hypo means less solutes compared to B. Since B has more compared to A, I would say B is hypertonic compared to A. Get it? Okay? If I compare B and C, which of the two is hypertonic? C, because this has more solutes, this has less solutes, right? So this is hypertonic compared to this, and this is hypotonic compared to this. Now if we look at, so hyper and hypo is based on how much solutes there is. The more solutes there are, the more tonic the solution will be. So tonicity is dependent on how much solutes there are. Alternatively, what you will see when we take when we compare hypo and hypertonic solutions, if we look at the amount of water, which of them has more water, a hypotonic or a hypertonic solution? Pardon? Hypotonic, right? So if this was hypotonic compared to this one had more solutes but less water. This one had more water. So hypotonic solutions have more water but less solutes. Hypertonic have more solutes and less water. Do you get that? That's only in osmosis. We are only talking about osmosis. This is only in osmosis, yes. We use these terms hyper and hypotonic when we talk about osmosis. Okay? Now let's see, we said that osmosis is the diffusion of water, and we take it for granted that the other substances, those solutes, were not able to pass. If they passed across, then equilibrium will be reached right through, okay? So if we had a beaker like this, and we had a membrane, a semi-permeable membrane, which only allowed water to pass through, and on this side we had lot of water, on this side we had half water and half solutes. So this would be hypertonic, this would be hypotonic, am I correct? Yes? I said osmosis is diffusion of water. What happens in diffusion? How do substances diffuse? Go from an area of high to low. Remember, diffusion is always along concentration or down the concentration gradient. So they always go from an area of high concentration to an area of low concentration. Think of it this way. In the lab, we did that today. We took a beaker of water and we put a little bit of food coloring dye. So it was concentrated in the center and then it started spreading out, right? It diffused from the center all over. And, you know, when you spray perfume on yourself, what happens? It a little bit goes in the air. It's sort of, in the beginning, it's very close to, you know, where you are, and then it diffuses all across the room. So the gas, those molecules kind of spread across. So it's always from an area of high concentration to an area of low concentration. In osmosis, the only difference is that we are talking about water. So it will be that water will go from an area of high concentration to an area of low concentration. So if you had this semi-permeable membrane and this side was labeled 1 and this side was labeled 2, which direction would water move? Would it move from 1 to 2? Would it go this way, or would it go from 2 to 1? It will go from 1 to 2, right? It will go this way because this is an area of high concentration of water and this is an area of low. So when it goes from here to here, this water level will come down and this water level will go up, and the volume will go up till the water is equal on both sides. The percentage or the amount of water is equal on both sides. Get that? Okay. Now, sometimes what happens is that suppose I was to close this beaker and I did not allow a volume change in the beaker. Then there would be kind of pressure exerted, isn't it? Right? If you had a beaker, and imagine this beaker was up here like this, and I did not allow the volume to go up. This side is trying to draw water towards it, but water cannot go towards it because, you know, I'm not allowing the water level to go up because the beaker is only that small. So the pressure that is generated in order to withstand that water getting in, that's called osmotic pressure. Okay? Pressure which is generated by solutes is known as osmotic pressure. So this side is generating the pressure because it wants to pull water towards it, but it can't because I'm not allowing that volume to go up. So we'll say that the more solutes you have, the greater the osmotic pressure. So another thing is, the more the solutes that you have, the greater the osmotic pressure. By this, it just means that if you have more solutes, there will be a tendency of water to try to go towards that area, right? Because more solutes means that solution is hypertonic. Water goes from hypo to hyper. That means hypertonic always is drawing water towards it. So the more solutes you have, the more the osmotic pressure.
I think that's why I'm confused. I don't know if I'm going to say anything wrong, but that's why I was confused because water's going from hypo
To hyper, yes.
But then osmosis in water is going from high concentration to low concentration.
High concentration of water to low concentration of water. So let me say this again. Okay, no, that's okay. So let's take a hypotonic solution and an hypertonic and define them. Hypertonic solution has more solutes, less water. Okay? Hypotonic solution has less solutes, more water. By definition, osmosis is diffusion of water, which means water will go from an area of high concentration to an area of low concentration, and high concentration of water. Okay? So it will go from hypo to hyper like this, right? Which means that hypertonic solutions are tending to pull water towards them, isn't it? Right? They draw water towards them. So the more the solutes you have, the more the water will be pulled towards it. solutes determine osmotic pressure. So if you have more solutes, your osmotic pressure will naturally be more. Have you followed that? Okay, if you have more solutes, your osmotic pressure will be more. And the way we look at this tonicity is that if we place a red blood cell, let's take a red blood cell, and we place it in a petri dish which has a solution in it which has the same amount of solutes and water as a red blood cell, we call that solution isotonic. So we put a red blood cell inside it. What is inside the cell is the same as what is outside. The cell will remain the same. Nothing will happen to the cell. Let's say we take a red blood cell and put it in a petri dish which has a hypertonic solution. So in other words, here's the red blood cell and the solution outside is hypertonic, right? That means the red blood cell is hypotonic compared to the solution outside. What will happen to the red blood cell? It will shrink because water will move out from the red blood cell, so this red blood cell will shrink, and that shrinking is known as crenation. Crenation. Let's take the opposite. We take a red blood cell and put it inside distilled water. So we put the cell inside, so the distilled water has no solutes at all, so the distilled water is hypotonic compared to the red blood cell. So the red blood cell is hyper and the distilled water is hypotonic. What will happen to the red blood cell? It will swell because water will move towards the red blood cell. It will get larger and larger. Since the cell membrane can only enlarge so much, after some time the red cells will rupture and they will die. Okay? So that's why in your body, whenever you're given fluids, most of the time we always want to make sure that you're given fluids which are isotonic to your body, so that we do not cause a change in the shape of the red blood cells. So that's the principle of osmosis, which is just diffusion, but in this case we are taking water. The third is what is called facilitated diffusion. As the name suggests, it's still diffusion, so still down or along a concentration gradient. But here, the molecules are really large. So like sugar molecules are very big, amino acids are big, and sometimes ions may not be able to pass through that easily. So in this case, they need something to help them along. So they need something called facilitation. So these things which help them along are known as carrier proteins. They still don't use energy. They just help them to pass from the outside to the inside of the cell. So it's always along a concentration gradient. Or even in some cases, if the concentration inside the cell is more, they might help it to go outside. They are known as carrier proteins. And lastly, under passive transport, so we said this was along concentration or pressure gradient. So far we've used, everywhere we used concentration. This was concentration of substances. Here we said concentration of water. And here again we used concentration of sugar, amino acids or ions, but using a carrier protein to go from higher to lower concentration. In parts of your body, especially for example the kidneys or even the blood vessels, in the kidneys and blood vessels, even in the kidneys it's the blood vessels. When urine is being formed, what happens is in the kidney you have a network of blood vessels like a big capillary plexus like this. And blood flows through this capillary plexus and passes out. And in this plexus of capillaries, the blood vessels have tiny little pores present. And next to it is a little sort of tube which lies next to this capillary plexus. So as blood flows through this, because of the presence of pores, some fluid passes out and comes into this tube, and then it travels through the kidney, and that's what comes out as urine. Now when blood is flowing through, it's fluid which is flowing through these capillaries. And when you think of fluid and think of a hose, for example. So if you attached a hose to a water faucet and you made tiny holes in that hose and you turn down the faucet, what happens? Doesn't fluid come out through those holes that you have? The same thing is happening here. So fluid is passing through this hose of capillaries. Because they are tiny holes, some of the fluid comes out and passes through and comes out of your body as urine. Fluid which passes through is under a pressure. So this is known as pressure gradient. So it's an area of higher pressure here and lower pressure here. That's why fluid is coming out. So wherever fluid passes through, that's known as pressure gradient. So here we are not looking at concentration of anything. We are looking at the fluid pressure, just like in your hose. As you turn on the faucet, the water in the hose is at a greater pressure than the pressure outside the hose. That's why water comes out through those little holes that you've made. So pressure gradient is also known as hydrostatic pressure. So this is also called hydro for water or fluid. So this is also known as hydrostatic pressure. And this is responsible for filtration. So wherever you have fluid flowing under great pressure, and this usually happens in blood vessels, and many blood vessels have small pores in them. Some of the fluid will pass out of the pores because there's more pressure inside the blood vessel than outside. And that results in, for example, in the kidneys, it results in urine being formed. In your blood vessels, it results in a little bit of fluid going out to bathe all the cells and provide nutrition. So this kind of pressure gradient is responsible for filtration. So filtration is nothing else but what is coming out of those pores. In fact, when you make coffee, what do? You put a little, the coffee, what is that thing called? Filter, yes, the coffee filter, exactly. So you put a coffee filter and you pour fluid through your thing, and that it's called a filter. Think of it. And what happens? Filtration, passing through that filter into your mug, and there you get coffee. So that's fluid which is passing through. So filtration is fluid passing through from an area of high pressure to an area of low pressure. And fluid pressure is also known as hydrostatic pressure, from the word hydro. Now let's look at these various modes of transport. So first we'll look at diffusion and osmosis.
Diffusion is the net movement of molecules down a concentration gradient. This process allows small molecules, such as oxygen and carbon dioxide, to cross the plasma membrane. Most polar molecules, such as sugars and proteins, cannot freely cross this lipid membrane. Although water molecules are polar, they are small enough to pass through the membrane freely. This special case of diffusion that involves the movement of water molecules across a membrane is called osmosis. If a molecule, such as urea, is added to one side of a membrane, it will not be able to diffuse across the membrane because it is both large and polar. Because of its polar nature, it will interact with other polar molecules, such as the water. This interaction reduces the number of free water molecules on the right-hand side. With fewer free water molecules on the right-hand side, there is now a net movement of water molecules down their concentration gradient to the side with the urea molecules. Because more water molecules are moving into this area than are leaving, the water level on the right side will rise. If the osmotic concentrations of two solutions are equal, the solutions are isotonic. However, when the solutions have unequal osmotic concentrations, the solution with the higher concentration of solutes is hypertonic, and the solution with the lower concentration of solutes is hypotonic.
So you saw in that how from hypo it went to hypertonic. Let's take a look at facilitated diffusion. So this is the outside of the cell, this is the inside of the cell. It's a large molecule which can't get through, so it has to pass through this. So here's a carrier protein. So notice how it goes in, attaches to this carrier protein, and this kind of facilitates or helps it and pushes it through and so it helps it to come down inside the cell. No energy is used in this case, and it's just making use of one of those integral proteins which is present in the cell membrane. And let's look at filtration.
Filtration is the first of three main processes in urine formation. Blood flowing from the glomerulus exerts pressure, and this glomerular blood pressure is high enough to push water and dissolved substances out of the glomerulus and into the Bowman's capsule. This fluid then...
When she's talking about glomerulus, she's talking about that capillary that I drew there, and the Bowman's capsule is nothing else but the part of the kidney which kind of receives that fluid, okay?
Becomes known as the glomerular filtrate. The wall of the glomerulus contains pores and is similar to a strainer. The diameter of the pores determines which substances flow through and which remain. The only blood constituents that do not move into Bowman's capsule are the larger molecules, such as blood cells and most plasma proteins, which exit the efferent arteriole. To summarize filtration, glomerular blood pressure causes filtration through the glomerular capsular membrane to create the glomerular filtrate.
Okay, let's look at this question. Yes, it will shrink, it won't swell. Remember, when we say hypertonic, we are comparing it. So the solution is hypertonic compared to the red blood cell. So the red blood cell, in this case, is hypotonic. So water will move out of the cell, making it shrink. Now, all this while we talked about active process. So let's look at, we talked about passive process. So now we are going to talk about processes which use energy. And in this case, energy, again, is what is ATP. One such example is in your body, you have these pumps called sodium-potassium pumps. Remember, I showed you all the method of that resting membrane potential where I told you the outside had more sodium ions and the inside had potassium. When nerve impulses come, the sodium moves inside and potassium moves outside, and that allows the nerve impulse to be transmitted. But after some time, you want this to come back to normal. In order for it to come back to normal, now some sodium ions have come inside, but they have to go outside, and there is already a lot of sodium ions outside. So they kind of have to go against their concentration gradient, which means it's like going uphill. So they need energy. In order to do that, you have these pumps which are called sodium-potassium pumps, which use ATP and allow you to do that. So let's take a look at this.
The sodium-potassium pump is an active transport mechanism. 3 sodium ions bind to the protein channel, and an ATP provides the energy to change the shape of the channel that in turn drives the ions through the channel. One phosphate group from the ATP remains bound with the channel. The sodium ions are released on the other side of the membrane outside of the cell, and the new shape of the channel has a high affinity for potassium ions, and 2 of these ions now bind to the channel. This binding again causes a change in the shape of the protein channel, and this conformational change releases the phosphate group on the cytoplasm side. This release allows the channel to revert to its original shape, and as a result, the potassium ions are released inside the cell. In its original shape, the channel has a high affinity for sodium ions, and when these ions bind again, they initiate another cycle. The important characteristic of this pump is that both sodium and potassium ions are moving from areas of low concentration to areas of high concentration. In other words, each ion is moving against its concentration gradient. This type of movement can only be achieved by the constant expenditure of ATP energy.
So when we looked at passive transport, remember we said it moved from an area of low concentration, an area of high concentration to an area of low concentration. So it was moving downhill, very easy. Somewhere where it was high, it had to come to an area which was low. In the case of active transport, for example, in the sodium-potassium pumps, we are moving uphill. So we are moving from an area of low concentration to an area of high concentration. So it's kind of uphill, which is why it needs energy and it needs these pumps to help it. The second type of active transport is where you form little round vesicles. Vesicles are like globules. So this is also known as vesicular transport. So in vesicular transport, you either bring substances from the outside into the cell, or you have substances inside the cell and you throw them outside. So when you throw it outside, it is known as exocytosis. Cytosis means in the cell, exo means outside. So throwing outside the cell. The second type of cytosis is where you take substances into the cell, and that's known as endocytosis. Endo meaning bringing into the cell. The three types of endocytosis are: one, where you actually take a large particle and you bring it into the cell. That kind of endocytosis is known as phagocytosis. Another word for phagocytosis is cell eating. It's almost as if the cell is eating something else. Macrophages, monocytes, and neutrophils are some examples of cells which are capable of doing this. Second type of endocytosis is, instead of getting an actual large structure into the cell, you just take fluid inside. So you get dissolved nutrients. So this is also known as, one example is also called cell drinking, or known as pinocytosis. Dissolved nutrients are brought inside. And a third type is where on the surface of the cell you have special receptors which will react only to the substances that they are receptors for, and usually these are hormones. So this is one way of getting hormones like insulin inside your body, inside your cell, so that insulin can carry out its function. Okay? So these are known as receptor-mediated endocytosis, meaning you have receptors, the substances attached to those receptors, and then they are brought into the cell. So let's take a look at some of these.
The substances used as fuel by single-celled organisms include other smaller cells, particles of organic material, and large molecules that cannot pass through the plasma membrane. Many single-celled eukaryotes use a mechanism called endocytosis to ingest such food particles. In this process, the plasma membrane surrounds and engulfs the food particle. Cells use three basic types of endocytosis depending on the size and nature of the material to be ingested: phagocytosis, pinocytosis, and receptor-mediated endocytosis. If the material the cell takes in is particulate, such as a bacterial cell or an organic fragment, the process is called phagocytosis. If the material is a liquid, it is called pinocytosis. Some types of molecules, such as low-density lipoproteins, LDLs, are transported across the plasma membrane by receptor-mediated endocytosis. These molecules first bind to specific receptors embedded in the plasma membrane. The receptor molecules are concentrated in an indented pit coated by the protein clathrin. When sufficient target molecules accumulate in the coated pit, the pit deepens, seals, and is incorporated into the cell as a coated vesicle. Exocytosis is the reverse of endocytosis. This process results in the discharge of materials from membrane-bound packages that migrate to the inner surface of the plasma membrane, fuse with the membrane, and then release their contents to the outside of the cell.
So here the same thing is shown over here in exocytosis. Exo means outside. The cell is pushing something outside, which is secretory vesicles. So if you look up here, this is a cell organelle called the Golgi apparatus. And in this animated thing, this is the inside of the cell, and this is the plasma membrane, this is the outside. So notice how the vesicles are coming, attaching to the plasma membrane, throwing out what needs to be thrown out, and then the rest of the vesicular wall gets incorporated into the plasma membrane. Same thing up here, and notice the Golgi body or Golgi apparatus makes up these vesicles which contain substances which need to go to the outside. So they travel and then they come to the surface of the plasma membrane where they attach to the surface and they throw their secretions outside. So this is called exocytosis, sending out of the cell. So it requires this movement, requires energy, so this is a means of active transport. The second type was endocytosis, when you take something in. So here also, this is another means of active transport. Again, you use ATP as energy. Endo meaning taking inside. So you could take solid particles, which could be bacteria, viruses, or anything else like proteins and so on. Or you could take liquid substances. And then another one is where you have specific receptors which are present on the cell membrane. Receptors are, imagine they are like stations which respond to only certain things. Usually you will find receptors in the body which react to enzymes or react to hormones. So this kind of endocytosis is known as receptor-mediated endocytosis, where you have these little spots or stations which we'll call receptors, and hormones specific to that receptor will come and get attached, and then the cell will take that inside and then use it for whatever it's needed for. So this is one example of receptor-mediated endocytosis. This one is where liquid molecules are taken in, known as pinocytosis, or also called cell drinking. And then this one is phagocytosis, where a solid particle is engulfed, and only certain cells are capable of phagocytosis. In order to phagocytose, the cell has to have a lot of cytoplasm in it. So usually the cells which are capable of phagocytosis have a lot of cytoplasm. Some examples are neutrophils, macrophages, monocytes, and as we go along, I will keep adding to that list. But for now, you can think of these three cells. They have a lot of cytoplasm. So what they do is, and you saw that little video, so that if there is a solid particle, the cell approaches that solid particle. This is the nucleus of the cell, and imagine here's a bacterium. This cell approaches this particle and sends out little cytoplasmic extensions which are known as pseudopodia. These are called pseudopodia, or meaning false feet. Pseudo meaning false, podia meaning feet. Then these pseudopodia come close to each other. This is the nucleus of the cell, and that particle is engulfed. So you can see these two pseudopodia have come close to each other like this. Over here, the cell membrane between them disappears, so this particle then gets into a secretory vesicle and the cell membrane closes off. And this then is attacked by lysosomes, which is a cell organelle which has digestive enzymes, and those kind of destroy the bacterium. So this kind of process is known as phagocytosis, or cell eating, and again a means of active transport. Okay. It's a means of active transport. So let's take a look at some questions up here. So we've done passive and active transport. So which of the following is not an example of simple diffusion? a neutrophil engulfing a bacterium, which is what I just showed you, which was phagocytosis, right? So that was active transport. Simple diffusion is passive transport. Look at all of this. Gas exchange in the lungs. Air, remember I told you in the lungs, the air sacs are very thin walls. Substances just diffuse across, gases. Popcorn odor permeating the room. So if you open up a bag of popcorn in the room, what happens? In the beginning, the odor is concentrated on the person who has the bag, and after some time, it kind of spreads all over, right? So this also is a kind of simple diffusion of odors. So from an area of high concentration, spreads to all of the surrounding area of low concentration. Sugar cube dissolving in water. You place a sugar cube in water, so it's concentrated in the middle, and you keep mixing, and then it dissolves. So everything now, the water is completely saturated with that sugar cube. So it's gone from an area of high concentration to reaching equilibrium, which is spread all over. Okay? Here, I just gave you this example of a neutrophil engulfing a bacterium, which is phagocytosis, which is not diffusion. This is active transport. Simple diffusion was passive transport. Okay? So we did plasma membrane and we did cell transport. Now let's look at the other constituent of a cell, which is the nucleus. This is very important for cell division. Like the plasma membrane, the nuclear membrane, it's surrounded by a nuclear membrane so that the genetic material, which is called DNA, which is present inside the nucleus, that is kind of contained. So that membrane is called the nuclear membrane. That also has pores and is selectively permeable, just allows some substances to pass through. Inside the nucleus, you have genetic material, which is DNA. When they are in single thread, when the DNA is in a single thread-like form, they are known as chromatids. It's an inactive stage. And then before the cell begins to divide, these single threads, what they do is, each one of these threads, they replicate or they duplicate themselves. So this single thread now becomes like a double-threaded structure, which is condensed and thick. That is known as a chromosome. Okay? So I'm going to repeat this. The nucleus contains DNA, which is the genetic material. In an inactive stage, this DNA is present in a very thread-like form and collectively known as chromatin. But if you examine each thread, it's very thin. That's known as a chromatid. Before cell division has to occur, these chromatids, the DNA in the chromatids, duplicate themselves. So a single strand like this will actually become two threads. So when it's two-threaded and it's kind of got thick, it becomes thick and short, then we call it a chromosome. They both are made of DNA. Inside the nucleus, we also have a smaller body, which is known as the nucleolus, which contains RNA and proteins. The nucleus is the bandmaster, if you can think of it, or, the conductor of the orchestra. So it gives orders, and especially for protein synthesis. So it's the one which gives orders, the nucleus, and that is through the DNA. The RNA is the one which takes orders. Then let's look at the cytoplasm, which is the activity center, where all of these small organelles are present and each one carries out a different function. So as always, you must know what is the function of that organelle, and automatically you should start thinking critically that if this is the function of this organelle, where would this organelle be, where would I see in more of it? So for example, let's look at mitochondria. Mitochondria are known as the powerhouse of the cell because they produce ATP, that's the energy source. So wherever cells need to produce a lot of energy in order to carry out their activities, those organs will naturally have more mitochondria. So take a look at this. Muscle cells automatically have a lot of mitochondria because, you know, when you exercise, when you move, you need oxygen, you need a lot of ATP to be produced in order to allow you to move. So muscle cells have a lot of mitochondria. Kidney and liver cells. Liver cells carry out a lot of metabolism. So they have lots of functions. Kidney cells also do a lot of filtration in order to make urine. So these three areas, you automatically expect that you will have many mitochondria in these cells compared to another cell, which another organ which may not do as much. This is not to say that only muscle, kidney and liver cells contain lots of mitochondria. There are other parts of the body, but I'm just giving you examples, okay? The next cell organelle is a very tiny organelle known as ribosomes. These synthesize proteins. They may lie free in the cytoplasm, or they may be attached to a set of tubules which is known as endoplasmic reticulum. So they may lie free, or they may be attached to something known as endoplasmic reticulum, and they make this endoplasmic reticulum look a little rough because they sit on the outside of the endoplasmic reticulum. So the membrane kind of looks studded with these little ribosomes, so it doesn't look smooth. So we'll see that that kind of endoplasmic reticulum which has ribosomes attached to it is known as rough endoplasmic reticulum. In either case, whether they are free or whether they are attached to endoplasmic reticulum, the function of ribosomes is protein synthesis. So therefore, in cells which have a lot of secretory function, and especially where those secretions are protein in nature, for example, cells which produce enzymes. Remember, enzymes are protein in nature. Some hormones are protein in nature. Such cells will have a lot of ribosomes, or they'll have this rough endoplasmic reticulum. In other words, something where ribosomes are attached to it. Okay? So gastrointestinal tract, the cells of the digestive system, produce a lot of enzymes which are protein in nature. So in order to produce that protein, they need ribosomes. So they may have free ribosomes, or they may have this kind of endoplasmic reticulum which has ribosomes attached to it. Okay? Yes. This is showing you, this part is just showing you one part of, if you just take a small part of this inner wall of the mitochondria, it's just showing you that and various enzymes which are there which help to produce ATP. Okay? So you can see that the mitochondria has an outer coat and an inner lining which is kind of thrown into little folds, has a lot of oxidative enzymes in it which help to produce this ATP. So here is that endoplasmic reticulum I was talking about. There are two types. So endoplasmic reticulum, the word reticulum means like a network. Endoplasmic, inside the cytoplasm. So this is a network of tubules which you can see up here. One of these has ribosomes attached to the outside, so they are sticking to the outside, giving it a rough appearance. So this is known as rough endoplasmic reticulum. Since ribosomes are outside and they manufacture proteins, one of the functions of this rough endoplasmic reticulum is to take that protein. The ribosomes manufacture it, so in effect the rough endoplasmic reticulum also manufactures it through the use of the ribosomes, and then it transports it through these tubules and actually passes it to another organelle lying close by, which we'll see, which is known as the Golgi apparatus. So the function of rough endoplasmic reticulum is to manufacture and transport proteins. It also manufactures membranes belonging to various organelles and the plasma membrane as well. So because of this manufacture and transport of proteins, it's naturally seen in a lot of secretory cells, because when cells secrete, most of the time the secretions are protein in nature. So like I said, I gave you an example. The digestive system was one, and pancreas is part of the digestive system. So here in the pancreas, which does a lot of secretion of enzymes, you expect to see a lot of rough endoplasmic reticulum. compared to the rough endoplasmic reticulum, there's one which is called smooth because it has no ribosomes attached to it. So if you look up here, this is also a series of tubules which have not been cut. Very smooth on the outside because they have nothing attached to it. So this is called smooth endoplasmic reticulum. Its function differs. Instead of producing proteins and transporting proteins, it is involved in processing fats. So lipid or fat metabolism. It also makes fats. And the liver is one very important organ which carries out lipid metabolism. So you see a lot of smooth endoplasmic reticulum in the liver. The liver also detoxifies drugs and carcinogens. So that's another reason why you see this smooth endoplasmic reticulum in the liver, because another action of the smooth endoplasmic reticulum is to detoxify drugs and carcinogens, cancer-producing substances. Smooth endoplasmic reticulum is also important for steroid hormone synthesis because steroids are fatty in nature. They have fat in them. So organs like the adrenal gland, which produce a lot of steroid hormones, hence have a lot of smooth endoplasmic reticulum. Another thing that smooth endoplasmic reticulum does is glucose is usually stored as a complex carbohydrate in us, in humans, and that complex carbohydrate is known as glycogen. But if the body needs glucose, and there is none available for it, it can break down this glycogen to glucose. And that breakdown is done by the smooth endoplasmic reticulum. And that breakdown is also done in the liver and skeletal muscle. So here you can see that how the liver contains smooth endoplasmic reticulum for many reasons. One is to produce and synthesize lipids, to detoxify drugs and carcinogens, to break down glycogen to glucose. So liver contains a lot of smooth endoplasmic reticulum. Another gland is adrenal gland because of the steroid synthesis, and skeletal muscle because even skeletal muscle glycogen is stored and is broken down. Okay? Let's look at another cell organelle which is known as the Golgi apparatus, or also called Golgi body. And if you pay attention to this picture, notice this is the Golgi body, again a series of tubules. But from these tubules, little round structures come off which are known as vesicles. And vesicles are basically tiny packets which contain substance inside them. So the function of the Golgi apparatus is to modify and package the material it gets from the rough endoplasmic reticulum. So here you can see rough endoplasmic reticulum, and the Golgi apparatus lies very close to it. So it gets these proteins from the rough endoplasmic reticulum, it goes into the Golgi apparatus which modifies it and then passes it out as secretory vesicles which it pushes to the outside. So by exocytosis, these proteins will go to the outside of the cell wherever needed. So again, the pancreas has a lot of Golgi apparatus because the pancreas secretes. Any cell which secretes a lot will have Golgi apparatus because finally the secretion is done through these small vesicles which are made up by the Golgi apparatus. The Golgi also forms certain vesicles called lysosomes, which are often known as suicidal sacs. So these lysosomes are also called suicidal sacs, because they have enzymes in them, acid hydrolase enzymes, which will digest any material which will be inside a vesicle. So remember I gave you that example of phagocytosis. So here was a cell which took in this foreign, this is the nucleus, took in this, so this became a vesicle, and then the cell kind of came like this. So then a lysosome will go close to it, will attach like this to the cell membrane, release its contents inside, and it will digest this bacteria which has gone in, and then the debris will be just passed out of the cell. So the lysosomes are formed by these Golgi apparatus. And as you can see, because they have digestive enzymes, their function is digestion. We have another type of enzyme, which are another type of vesicle, small structures like this, which are known as peroxisomes. They detoxify substances. So their enzymes are what are called oxidase and catalase enzymes, usually meant to detoxify drugs, alcohol. So the liver, which we saw was one of the areas where all this happens, has a lot of these peroxisomes. Kidney also has some of them, of these peroxisomes. Compared to other cells, it has more, because it helps to detoxify many of these substances which get into the body and need to be removed. So you may have heard of free radicals. So much is talked about free radicals, which kind of are harmful to your body. So these peroxisomes are the ones which kind of attach to these free radicals and prevent them from remaining in the cell. So that's how they help to detoxify. So let's take a look at this question and read the question carefully. It says, intensely biosynthetic secretory cells, such as neurons, would be expected to have great amounts of compared to other cells. Okay, the correct answer is rough endoplasmic reticulum. And here's why you're going to think of it. Look at the word. The operative word is secretory cells. It doesn't matter which type of cell it is, whether it's a neuron or a muscle or liver, but I'm giving you a hint, secretory cells. And I told you, most of the time these secretions are protein in nature. So which is the organelle which produces proteins? Rough endoplasmic reticulum, right? Lysosomes are suicidal sacs. So they are present when something has been taken into the cell and that needs to get digested. I haven't given you anything in the question which suggests that. I haven't said that, which cells which tend to phagocytose substances and what will they have. Instead, I'm telling you something is going out of the cell. Peroxisomes are ones which, again, detoxify. Again, I'm not talking about detoxification, I'm talking about secretion. So here you're going to think of what was connected to secretion. And I told you all earlier that secretions are usually protein in nature. So which of these produces proteins? It would be rough endoplasmic reticulum. Now let's say you had another choice called Golgi apparatus up here, and you could choose two. So you can see that any cell which is secretory would have rough endoplasmic reticulum to produce the protein. It would also have Golgi apparatus to make that package that protein, put it into little vesicles and push it out of the cell. Okay? So secretory cells therefore have a lot of rough endoplasmic reticulum and a lot of Golgi apparatus. The next organelle is something called the centriole, which is really important for cell division because it lays down what we'll see, which is known as the mitotic spindle. So here is a centriole which is shown. It also is sort of anchors cilia and flagella, which I showed you, which were modifications of the cell membrane. So it anchors cilia and flagella to the cell. So imagine if you have a cell like this and you have cilia up here, centrioles anchor these cilia to the cell. So that's another function of centrioles. Cilia and flagella, yeah. The attached end, this end, there's a free end of the flagellum and cilia. Then we have elements which are known as cytoskeletal elements. If you look at the word, it's like the skeleton of the cell. So these are, you get very, very tiny tubules which are known as microtubules. They kind of make the skeleton, the inner skeleton of the cell. They also help to direct how the spindle should be formed. And then especially in muscle cells, we have tiny thin filaments present, which are known as microfilaments. And when we do muscle cell, we'll be talking about them. These microfilaments in muscle are known as actin and myosin. And they have contractile properties. So we see these two in cells. And when we discuss muscle, we'll talk a little bit more about these microfilaments. Myosin. So those were all the cell organelles that we looked at. Now let's take a look at how a cell divides. So there are two parts to cell division. One is nuclear division and one is cytoplasmic division. Because both the nucleus and the cytoplasm have to divide in order for the cell to duplicate itself, for one parent cell to make two daughter cells. So if you have a cell like this with a nucleus, you want to end up getting two cells like this. So not only does the nucleus have to divide, the cytoplasm also has to divide. Nuclear division is known as mitosis, and it has different stages. These are the stages of mitosis. Cytokinesis is cytoplasmic division. There are no stages to cytokinesis. It starts during one of these mitotic phases. It begins during one of these mitotic phases. So let's look at some of the things that happen. So imagine you have a cell like this, and I'm going to take a simple cell, and let's say this is the nucleus of the cell, and this cell has 2 chromosomes. Okay, so just for the sake of color, I'm going to put, it has 2 chromosomes. That's one pair of chromosomes, right? And you want this to divide and form 2 cells, and your 2 cells have to be identical. So you've got to get 2 cells like this, with a nucleus inside, and these 2 daughter cells also have to have a pair of chromosomes or 2 chromosomes. Right? Now if you began with this cell and you didn't do anything, and this cell if the nucleus divided, it will divide right in the middle. It could divide right in the middle like this. So over here, what would you get? One cell would get a blue chromosome and the other one would get a red chromosome, right? If we did it this way. Or if we did it across, then we'll get half the DNA in one cell and half the DNA in another. So we are not getting identical cells. So in mitosis, we need identical cells. So what is one of the things that the cell does? So if we take the nucleus of the cell, and let me just draw the nucleus. I'm only going to draw the nucleus, okay? So here's the nucleus of the cell, and let's say we started out with 2 chromosomes like this. What if we duplicated the DNA? We cannot change the number of chromosomes, because the number of chromosomes that a species has determines what that species is. So human beings, for example, have 46 chromosomes or 23 pairs. If we change 46 into 92, then we become a whole new different species. So we cannot change that, right? However, DNA is capable of replication. So imagine if this DNA, this chromosome here, instead of consisting of just one thread, the DNA duplicates, and this is now made up of 2 threads, but the 2 threads are attached to each other. So it's still 1 chromosome. And we do the same thing here for the red one. So the DNA, before cell division begins, the DNA duplicates itself. And each chromosome now consists of 2 threads instead of having 1 thread. So we still have 2 chromosomes, right? Now, if these 2 were to line up one below the other, so if they were to line up like this, one below the other, and now we divide it right through, can you see that we'll get 2 cells which will be exactly alike the parent cell. So this is the nucleus of one, this is the other one. So we began with this. We started out this way. Then we had to duplicate the DNA. So this was the parent cell. And these are the 2 daughter cells. And I'm only showing you the nucleus. So can you see that the 2 daughter cells also now have 2 chromosomes, right? Can you see that? So that's why the DNA has to duplicate. And I give a not so good example. It's like, for example, if you have a fruit which is 5 pounds and you want to get 2 fruits which are exactly the same as this. So you increase the weight of that fruit to 10 pounds and then you can divide it into 5 and 5, right? Something like that. But this is not weight, but it's the amount of DNA, okay? So we'll say that if the amount of DNA to begin with in this cell, because it contains 2 threads, say the amount of DNA is called, not the number of chromosomes. The chromosome number is 1 pair or 2, but the amount of DNA also we can say is 2n. If I just use the word n as a way of determining how much DNA is present, like that is how much DNA is present inside all of this. Because there are 2 threads, I'll say it's 2n. So as cell division is going to progress, during a phase which is known as interphase, the DNA duplicates. So now the amount of DNA has doubled. Can you see? So the chromosome number is still 2, but the amount of DNA is 4n. Have you seen that? Can you see that? Then as division occurs, finally when we get the 2 daughter cells, the chromosome number is 2 and the amount of DNA also is 2n between the 2 cells. Have you followed this concept? What you need to know is that whatever amount of DNA you start with, whatever a normal cell has, before cell division, that amount will double. And then at the end of the cell division, the amount will be the same as parent cell. So I could call this 4n, I could call 8n, I could call it anything. And that 8 will then become 16. If I said it was 4, it would become 8. So it just doubles. If I just said this was n DNA, this would be 2n. Okay? So if I just said that I'm just using that there is n DNA in a normal cell. Before cell division, what would be the amount of DNA? The n would become 2n, right? Here I just said 2n and it became 4n. In other words, it doubles the amount. But the chromosome number remains the same because these threads are still attached to one another. So you don't count this as 1, 2, 3, 4 chromosomes. You still count it as 1 chromosome and 2 chromosomes. You cannot change the chromosome number, but you can change how much DNA there is within each chromosome. Have you followed? Okay? So here, that's the phase just before cell division occurs. The DNA duplicates or replicates, so it becomes double. And the centrioles also, there's 1 centriole, you get 2, so that 1 can go to either end. And then the actual nuclear division starts. The first phase is called prophase. It's the longest phase. Here, when I say chromosomes are formed, that means that the 2 threads, they start condensing and the chromosomes become shorter and they get really condensed, because if they remained long and very thread-like, they could snap off during cell division, because the chromosomes are being pulled, as you will see. So if they remain short, then there are less chances of them breaking off. So chromosomes are formed because that DNA condenses and they become really thick. We'll also see how a mitotic spindle is formed. The spindle is like a framework on which the chromosomes can attach themselves. And obviously the nuclear membrane has to disappear if you've got to pull the chromosomes out and, get into daughter cells. The second phase is called metaphase, which we'll see. And the way you're going to remember, the chromosomes come on the center of the cell. They meet at the center of the cell or the equator of the cell. Then we'll see that the chromosomes kind of get pulled apart. So that is called anaphase. That's the shortest phase. The centromeres are those little things I showed you up here. When the two threads are attached, this part here is called a centromere. This is what is called a centromere. So the splitting occurs right through the centromere so that one thread goes to one side and one goes to the other side. At this phase, during the phase of anaphase, this cytokinesis begins. So a little furrow appears in the cytoplasm, and that furrow is known as the cleavage furrow. At telophase, we will see that the chromosomes now have split and gone to either ends or opposite poles. They were thick and coiled, now they uncoil, and then they start forming those single threads. The nuclear membrane reforms, the spindle disappears, the nucleolus reappears, and you're getting back to kind of coming to a proper cell. And then cytokinesis is finished or completed at telophase. So at telophase, the cytokinesis is completed and two daughter cells form. Okay? So we'll look at all of these stages. So this was cell division. In response to cell division, we also have something which is known as apoptosis. This is programmed cell death that occurs in our body. So as cells are dividing and making more and more cells, there are also areas in the body where cell death is occurring and cells are destroyed because you no longer need them. And that is programmed cell death. Okay? So let's look now at the phases of mitosis, and we'll also look at cytokinesis side by side. So here is the first interphase where you can see the chromatin inside the DNA. It's kind of duplicated and it's like a big network up here. The nuclear membrane is going to sort of start disintegrating in the next phase. Here you can see the centrioles, they'll duplicate. This then goes into the phase of prophase. So here the nuclear membrane is going to begin to disappear. Look at the chromosomes. Instead of being thin and thread-like, now they are thick. Each is made up of two threads, as you can see. The centrioles go to the opposite end and they are forming this mitotic spindle. Then these chromosomes, you want to bring them to the center of the cell, which is called the equator of the cell. So at this point, they are beginning to come to the equator. The nuclear membrane is disappearing. Look at this spindle. It's nicely formed. So this is prophase. The next phase is metaphase, where the cells meet, where the chromosomes meet at the midline. That's the way you can remember. So notice how the chromosomes are all lined at the midline up here. And they are attached to the spindle, and these are the rays of the spindle. And these rays attach to this centromere right here, that central little thing where those two threads are attached. The next phase is anaphase. The way you can remember anaphase is because it looks like an A. If you look here, it looks like an A. So in anaphase, you get the chromosomes being pulled because this spindle at either end, it contracts. So it begins to pull on the chromosomes. And they split at that centromere. So one thread goes to this end and one thread goes to this end. So you can see that, right? So you can see them moving away from each other. At this anaphase, notice a small cleavage furrow begins to form. So cytokinesis is beginning to occur at anaphase. This phase is telophase, where the chromosomes now go to the ends of the poles. The nuclear membrane will start forming, and cytokinesis will get completed, and then you'll end up with two daughter cells, and which are identical to the parent cell that they began with. Okay, so each will have the same amount of DNA and the same number of chromosomes. So let's take a look. Yeah, you had a question? No, they can't float off because you have this mitotic spindle which keeps them in place. That's why you need the spindle. It's a framework. It's a scaffolding on which these chromosomes can attach themselves. Okay? So let's take a look at this. So this doesn't have any audio in it, so just watch. So let's go back. So this is a chromosome with 2 threads. That's the centromere. These 2 threads are known as chromatids. So this is prophase. Centrioles at either end. Nuclear membrane will disappear. Chromosomes get real thick. So they go to either pole. Metaphase, they meet in the middle. Anaphase is going apart. So they move apart. They don't go to the ends at all. Telophase is when they go right to the ends, and now the cytokinesis gets completed and you end up with 2 daughter cells which are identical to the parent cell. And during the interphase, before cell division occurs, that's when remember the DNA duplicates so that you can end up with the same amount of chromosomes, the same amount of DNA as the parent cell. So let's say you got a question like this. In which of the following cells will you not see spindle formation? Now, these are not arbitrary cells that I've thrown here. This is because sometime when we were discussing cells, I mentioned something about one of these cells, something specific. So that is important, and based on that, you're going to kind of choose your answer. Very good. Most of you got this right. Yes, red blood cell. Red blood cell does not have a nucleus. So if it doesn't have a nucleus, it obviously cannot undergo cell division because the mitosis part of cell division will not occur. So there will be no spindle formation. I didn't give you anything else about any of these other cells. I specifically mentioned red blood cell not having a nucleus. So therefore, the answer is red blood cell. All of these have a nucleus. They all do tend to divide normally, and you will see a spindle. Okay? So that's how you're going to approach questions like this. You have clues in your notes or told to you in class, and you just got to kind of connect the dots. So always read the question very, very carefully because there'll always be some operative words in that. When you get a quiz question wrong, go back and read and see why you got it wrong and look for the correct answer in your notes or in your PowerPoint, or also go back and listen to the video if necessary.